Replication. What is DNA replication? DNA replication is the process by which a molecule of DNA is duplicated. When cell divides, it must first duplicate its genome so that each daughter cell winds up with a complete set of chromosomes. DNA replication is the process by which DNA makes a copy of itself during cell division. DNA replication is an essential part of cell division and the growth of organisms. The process of DNA replication uses a strand of DNA as a template to create new strands of DNA. The process of DNA replication is vital for cell growth, repair, and reproduction in organisms. Why DNA replicate? DNA is the genetic material that defines every cell. Before a cell duplicates, and it's divided into new daughter cells through either mitosis or meiosis. Biomolecules and organelles must be copied to be distributed among the cells. DNA found when the nucleus must be replicated in order to ensure that each new cell receives the correct number of chromosomes. DNA structure. DNA or deoxyribonucleic acid is a type of molecule known as nucleic acid. It consists of 5 carbon, deoxyribose sugar, a phosphate, and a nitrogenous base. Double-stranded DNA consists of 2 spiral nucleic acid chains that are twisted into a double helix shape. This twisting allows DNA to be more compact in order to fit within the nucleus. DNA is packed into tightly coiled structure called chromatin. Chromatin condenses to form chromosomes during cell division. Prior to DNA replication, the chromatin loosens, giving cell replication machinery access to the DNA strands. Preparation for replication. Step 1. Replication fork formation. Before DNA can be replicated, the double-stranded molecule must be unzipped into 2 single strands. DNA has 4 bases called adenine, thymine, cytosine, and guanine that form pairs between the 2 strands. Adenine only pairs with thymine and cytosine only binds with guanine. In order to unwind DNA, this interaction between base pairs must be broken. This is performed by an enzyme known as DNA helicase. DNA helicase disrupts the hydrogen bonding between base pairs to separate the strands into a Y shape known as the replication fork. This area will be the template for replication to begin. DNA is directional in both strands, signified by 5 and 3N. This notation signifies which side group is attached to the DNA backbone. This 5N has a phosphate group attached, while the 3N has a hydroxyl group attached. This directionality is important for replication as it only progresses in the 5 to 3 direction. However, the replication fork is bidirectional. One strand is oriented in the 3 to 5 direction, leading strand, while the other is oriented 5 to 3, lagging strand. The two sides are therefore replicated with two different processes to accommodate the directional difference. Replication begins. Step 2. Primer binding. The leading strand is the simplest to replicate. Once the DNA strands has been separated, a short piece of RNA called a primer binds to the three ends of the strand. The primer always binds as the starting point for replication. Primers are generated by the enzymes DNA primase. Step 3. Elongation. Enzyme known as DNA polymerases are responsible for creating the new strands by a process called elongation. There are 5 different known types of DNA polymerases in bacteria and human cells. In bacteria such as E. coli, polymerase III is the main replication enzyme, while polymerase first, second, fourth, and fifth are responsible for error checking and repair. DNA polymerase III binds to that strand at the site of primer and begins adding new base pairs complementary to the strand during replication. In eukaryotic cells, polymerase alpha, delta, and epsilon are the primary polymerase involved in DNA replication. Because replication proceeds in the 5 to 3 direction on the leading strands. The newly formed strand is continuous. The lagging strand begins replication by binding with multiple primers. Each primer is only several bases apart. DNA polymerase then add piece of DNA called Okazaki fragments to the strand between primers. This process of replication is discontinuous as the newly created fragments are disjointed. Step 4. Termination. Once both the continuous and discontinuous strands are formed, an enzyme called exonuclease removes all RNA primers from the original strands. These primers are then replaced with appropriate bases. Another exonuclease proofreads the newly formed DNA to check, remove, and replace any errors. Another enzyme called DNA ligase joins Okazaki fragments together forming a single unified strand. The ends of the linear DNA present a problem as DNA polymerase can only add nucleotides in the 5 to 3 direction. The ends of the parent strands consist of repeated DNA sequence called telomeres. Telomeres act as a protective caps at the end of chromosomes to prevent nearby chromosomes from fusing. A special type of DNA polymerase enzyme called telomerase catalase. The synthesis of telomere sequence at the end of the DNA. Once completed, the parent strand and its complementary DNA strand coils into the familiar double helix shape in the end Replication produces two DNA molecules, each with one strand from the parent molecule and one new strand. DNA enzymes. DNA replication would not occur without enzymes that the catalase virus steps in the process. DNA helicase unwinds and separates double-stranded DNA as it moves along the DNA. It forms the replication forks by breaking hydrogen bonds between nucleotide pairs in DNA. DNA premise, a type of RNA polymerase that generates RNA primers. Primers are short RNA molecules that act as templates for the starting points of DNA replication. DNA polymerase synthesize new DNA molecules by adding nucleotides to leading and lagging DNA strands. Topoisomerase or DNA gyrase unwinds and rewinds DNA strands to prevent the DNA from becoming tangled or supercoiled. Exonucleases, group of enzymes that remove nucleotide bases from the end of the DNA chains. DNA ligase joins DNA fragments together by forming phosphodiester bonds between nucleotides. DNA replication is the production of identical DNA alleles from a single double-stranded DNA molecule. Each molecule consists of strand from the original molecule and a newly formed strand.